At the end of this animation, you should be able to explain the role that epigenetics plays in tissue identity and the development of disease, distinguish between active and repressed chromatin, identify some of the enzymes that are responsible for modifying histone tails, explain how histone modifications influence gene expression, explain how the same chemical mark placed on a histone tail can signal for two very different events depending on the context of the mark. Epigenetic pathways regulate gene activity in a heritable manner without changing the primary nucleotide sequence of DNA. By turning genes on or off, epigenetics determine which genes are expressed, and these expression patterns are maintained through cell division. When cells of discrete tissues in the mammalian body divide, it is epigenetics that maintains their tissue identity. For instance, when your skin cells divide, they remain skin cells. Tissue state is controlled by the on or off status of sets of genes, thereby ensuring that when a skin cell divides, it does not turn into a hair cell or a bone cell. Most tissues in the human body contain exactly the same genetic blueprint. Tissue identity depends on which genes are turned on and off. Gene expression also plays a role in the development of disease. Cancer is an example where too much or too little epigenetic activity can be problematic. For example, epigenetic dysregulation can lead to repression of tumor suppressor genes or the activation of oncogenes. Both conditions contribute to the initiation and progression of cancer. Let's explore the process of turning genes on and off. Not surprisingly, it begins with DNA, which holds all the genetic information needed to create the entire organism within a single cell. In the cell, DNA is wound around proteins known as histones to form chromatin. Chromatin can be either active or repressed. Active chromatin is loosely arranged, much like pearls on a necklace. This conformation of chromatin is sometimes referred to as beads on a string. An open conformation allows DNA transcription to readily occur. DNA is transcribed into RNA when RNA polymerase binds the chromatin and transcribes RNA from the DNA template. The more active the gene, the more RNA is produced. RNA is the product of an active gene. When chromatin is repressed or inactive, it becomes tighter and more compact. RNA transcription ceases, and the functional activity of the gene is quenched. How is this complex gene regulatory network controlled? That's where epigenetics comes into play. Remember that chromatin is comprised of DNA that is wound around histone proteins. A single unit of DNA wrapped around the histone core octamer is called a nucleosome. Histones have tails that protrude out from the nucleosome structure. Enzymes place chemical modifications, or marks, onto amino acids on the histone tails to regulate gene expression. Some marks activate a gene, other marks repress a gene, while still others can either activate or repress a gene depending on where the mark is placed. Thus, the same chemical mark could signal gene activation or repression. The location or context of the mark makes a difference. While dozens of different histone tail modifications are possible, in this animation we will focus on just two important examples: acetylation and methylation. Acetylation of amino acids on histones generally correlates to gene activation. Acetylation reduces the positive charge of histone tails and repels them from the negatively charged DNA backbone in the packed nucleosome. This modification causes a slightly more open chromatin structure. Lysine and arginine amino acid residues on histone tails can also be methylated. Methylation is more complex than acetylation. Its correlation to gene activity depends on the context of where the mark is placed. For instance, when methylation marks are placed on lysine 4 of the tail of histone H3, usually this correlates with actively expressed genes. However, when methylation marks are placed on lysine 9 or lysine 27 of the tail of histone H3, genes are usually repressed. Epigenetic regulation is established and maintained by three major classes of proteins, or enzymes, that play distinct but intertwined roles during this process. Readers bind either active or repressive marks on chromatin. Writers place either active or repressive marks onto chromatin. erasers remove marks from chromatin. These enzymes perform their job simultaneously, meaning that an active mark might be erased at the same time that a repressive mark is placed, and vice versa. How these enzymes work together is an active area of research. What is known is that epigenetic processes have the ability to turn on and off genes. The regulatory process is dynamic and reversible. That is, repressive marks can be removed, active marks can be added, and RNA production can resume, thereby converting repressed chromatin into active chromatin. The flexible nature of this process is critical for many aspects of early human development and life. The main thing to remember is that specific marks on the histone tails play a role in turning a gene on or off. On means that RNA is produced. Off means that no RNA is made. These marks help hold tissue identity and can be maintained through cell division.